Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So we were discussing about immunology, and in today's lecture we are going to discuss about pattern recognition receptors. So this lecture is kind of introductory lecture about pattern recognition receptors. So we are going to discuss about how this idea evolved over the time, right? So we are going to discuss about innate immunity, its non-specific defense, its broad spectrum, its broad specificity, as well as how Charles Janeway, he gave us an idea about this PRRs, the PAMPs, DAMs, development of adaptive immunity. We will also talk about Spätzle-Toll-cactus pathway, which was given and which gave us evidence about this PRRs. So we are going to talk about that. So this is kind of introductory lecture, and then we will be discussing about the types of PRR in detail in separate lectures. So this is just going to give you an idea about what the pattern recognition receptors are and how the researchers developed this idea over the time. So before that, let's just revisit a few concepts. The immune system, so since the start of this immunology series, we discussed that we have two types of immune systems, the innate immunity versus the adaptive immunity. So the immune system is the biological fortress of our body, and it protects the organism from the pathogens such as bacteria, viruses, fungi and parasites, right? So it is broadly categorized into those two types, where the adaptive immunity is specific and it involves the memory generation, whereas the innate immunity, unlike the adaptive immunity, it is evolutionary ancient, it is non-specific, and it acts as the first line of defense. So it is characterized by a non-specific way of working, but over the time, due to the theoretical framework given by Charles Janeway, which was given in 1989. So Charles Janeway gave a theoretical framework which actually gave the idea of innate immunity. It's not exactly non-specific, but it has a broad specificity. So it has a broad spectrum of the patterns to which the innate immunity is going to react and mounts its responses. So rather than calling it innate immunity as non-specific, we actually refer at as that it has a broad specificity, meaning that it can recognize conserved molecular patterns of pathogens. We offer broad spectrum of those molecular patterns rather than unique antigens. So I also discussed this in the first lecture where we discussed about overview of host defenses, that there is something called broad specificity and the narrow specificity. Okay, so this wide range of specificity actually refers to a phenomenon where the receptor molecules, they are able to recognize a broad category of molecules such as the lipopolysaccharides or the beta glucans found on the fungus, right? Whereas the antibodies, on the other hand, they are really specific for certain antigen, right? So the interaction, the antibody antigenic interaction is very specific, it is highly specific. Whereas the interaction of those receptors, few receptors, the ones which we are discussing in this lecture, the pattern recognition receptors, they are broadly specific. So they can recognize the lipopolysaccharides, the peptidoglycans, they can even recognize the beta glucans. So they are recognizing a broad spectrum of molecular patterns, right? So that is why, and this, they are the part of innate immunity, and that is why we say that innate immunity has a broad specificity. So this foundational breakthrough of in our understanding of immunity, it came not from the mammals but from the invertebrates, the Drosophila melanogaster. Okay, so it is really interesting how these model organisms gave us a whole new idea and opened a whole new perspective of looking towards the innate immunity. So we are going to, so today's discussion is going to revolve around those two foundational breakthroughs, those two papers. The first one is approaching the asymptote. It was given by Charles Janeway in 1989, whereas the second one actually gave the evidence about whatever was the theoretical framework was being discussed by Charles Janeway in that Cold Spring Harbor Symposium. So the evidence was given in this paper just few years later in 1996 by the Jules, Hoffmann and the colleagues, where they discovered that this Spätzle-Toll-cactus pathway, okay, which actually has a role in embryonic development of Drosophila. It also is important for the defense against the fungus in the Drosophila. Okay, so we are going to discuss about how those two important seminal papers published by different groups of the researchers, how it gave us an idea about the innate immunity as well as the advancement or the initiation of the adaptive immunity. So, unlike vertebrates, Drosophila lack adaptive immunity. Okay, so there are no antibodies, no memory B cells, or no T cells in the Drosophila, yet it survives this microbe-rich environment. So, if you have ever worked with Drosophila melanogaster, you would know that it is very so the rearing of the Drosophila, or let's say the crosses that we do with the Drosophila melanogaster, it's very easy. So that is why it is considered as a model organism, right? So it's this most beloved model organism, Drosophila melanogaster, which is really easy to handle, okay, in the lab environments. Still, it's so when it's out there in the nature, it survives this microbe rich environment, even though even though it lacks this adaptive immunity. So this only suggests that the innate immunity which Drosophila has, it is very sophisticated. So it is going to mount sophisticated responses against the pathogens so that the elimination of pathogens will be seen. So innate immune responses or innate immune mechanisms in the Drosophila, they were studied by those researchers, the Bruno Lemaitre and the Nicholas, okay, and the Jules Hoffmann, and that gave kind of evidence for the theory, for the the theoretical framework given by Charles Janeway. So whatever Charles Janeway discussed in his essay in the Cold Spring Harbor Symposium back in the 1989, it was considered speculative. But this paper, which was published few years later, it gave us the evidence about this innate immunity being having broad specificity. So in Drosophila, this toll pathway which responds to the fungal as well as gram-positive bacterial infection, it revealed the activation of toll receptor and how it is important for the defenses against the fungus and the bacteria. So we will be discussing about the pathway, this Spätzle-Toll-cactus pathway in detail, and how it resembles to the NF-kappa B pathway in the mammals. So this is a task for you. If you don't know about NF-kappa B pathway, please learn about it and take a look at the similarities between this pathway, toll pathway, and that pathway. Okay, so you will see a lot of similarities, and you will also be able to discriminate between those two pathways, right? So we will discuss about this in a while, but before that, I'd like to tell you about what Charles Janeway had discussed in his essay. So he gave us the theoretical vision about the molecular precision of the innate immunity. So this Charles Janeway's hypothesis, he proposed that the initiation of adaptive immune responses, it is not spontaneous or random, but it depends on the evolutionary conserved receptors of the innate immunity, which he termed as the pattern recognition receptor. So see, the adaptive immunity is not independent. It is not going to mount its responses spontaneously, but it needs help of innate immunity. So first, innate immune system is it initiates its responses. So first, innate immunity gets activated, and then adaptive immunity is going to get activated in the vertebrates. So this theoretical framework as well as this particular paper which came later, both of those, they formed, they breached the gap between the our understanding of the immune system of invertebrates as well as the immune system of vertebrates. So the ideas of Janeway's, which were considered speculative, they had the evidence, and they had a spectacular molecular realization through this paper. So this paper actually it gave it discussed about this pathway of Spätzle-Toll-cactus pathway, its role in the antifungal immunity. So this toll receptor pathway in the Drosophila, this is actually important for the embryonic dorsoventral patterning. Okay, and it is also, so this was previously known for only this purpose. So it serves the it is important in embryonic development, the dorsoventral patterning of Drosophila. But then it found then researchers found in this paper that it is critical for the antifungal immunity as well. So this was first genetic demonstration of PRR like pathway. So the idea of PRR was given by Charles Janeway. Okay, I am not sure whether I am pronouncing it correct or not. And this idea, which was given by Janeway, it was, its first genetic demonstration was given by the Jules Hoffmann and his colleagues. So they also received Nobel Prize with Ralph Steinman. So I hope you remember that Ralph Steinman was a person who received his. I mean, the Nobel Prize was discovered just 3 days later his death. So he received the Nobel Prize posthumously. So his wife actually received the Nobel Prize. So Ralph Steinman, Jules Hoffmann, and someone else, I don't really recall the name. They those researchers, they received the Nobel Prize back in the date for their discovery about those stuff. So the this this paper, this essay is legendary, and this particular research paper proves that essay. So those are really foundational pieces in the history of the immunology. So here in this paper, the toll receptor pathway, which is important for antifungal immunity, it was discussed. So this is a PRR like pathway, pattern recognition like pathway. So in a multicellular organism like Drosophila, which doesn't have the adaptive immunity. So their study revealed that the fungal infection, whenever there's fungal infection, a proteolytic cascade will be activated, which turns a protein known as Spätzle protein in its active form. And once the Spätzle is activated, it is going to result into an intracellular signaling cascade, which will be important for dealing with fungus. So this Spätzle, it is a cytokine like molecule. Okay, so the cytokines are found in humans, right? So it is cytokine like molecule which binds, when once it is activated, it binds to the receptor known as toll receptor. Okay, so let's discuss about this pathway in detail so that you'll have an idea about the pattern recognition receptors. Okay, so let's discuss about Spätzle. Toll cactus pathway. Okay, so first of all, whenever there's fungal infection, so the microbial components of fungus, such as the beta glucans, which are present in the fungus. So this beta glucans, okay, beta glucans, okay, they will be recognized by the serine proteases and the accessory molecules which are present in the Drosophila. So there are some some accessory proteins such as GNBPs, okay, GNBP3. So these are these are the serine proteases, means they are going to do the proteolytic cleavage of certain molecule. So what do they cleave? So those enzymes, they cleave a molecule known as prospadzole, which is the inactive form of spadzole protein. So they are going to cleave the prospadzole, and once they cleave this prospadzole, it will be converted into its active form, spadzole. So once this spadzole protein matures, it resembles to cytokine, and that is why spadzole is also referred as cytokine like molecule. So the activation, so activation of spadzole, okay, from the prospadzole form to the mature spadzole form will be done by those serine proteases, so GNBPs or the Persephone. So they are the ones which are going to work on those prospadzole and will convert them into the active form. So once this active spadzole is activated, it is going to go and bind to the toll receptors which are present over the surface of the cells. So those toll receptors, the extracellular domain of the toll receptor binds to this spadzole, and once it binds to spadzole, it gets dimerized, and this dimerization of the toll receptor, it is going to activate the intracellular signaling pathway. So spadzole binds to toll receptors. Okay, So toll receptors get dimerized, and once they get dimerized, they are going to get activated and they are going to recruit certain adapter proteins such as MyD88, okay, which will be, which will activate certain kinases, and it will culminate in the phosphorylation and the degradation of cactus. So this will result into the degradation of cactus. Okay, so this cactus protein is inhibitor kappa B like molecule. Okay, so if you remember NF kappa B pathway, so inhibitor kappa B inhibits the NF kappa B. So cactus is inhibitor just like inhibitor kappa B. So once it is degraded now, so if it is degraded, now there will be release of a protein known as dorsal or DIF. So the dorsal and DIF, degradation of cactus will release dorsal and DIF, which is going to enter into nucleus, and it is going to drive the transcription of AMPs such as drosomycin. Okay, so antimicrobial peptides such as drosomycin, which are antifungal. So drosomycin has antifungal property, and this is how the fungus infection will be tackled with the help of Spätzle-Toll-cactus pathway. I hope this pathway is clear to you all. So this pathway, which was previously known for the and dorsoventral patterning in the Drosophila, the embryonic dorsoventral patterning in the Drosophila, it was now known for antifungal, for the potent antifungal response in the adult Drosophila. So this was a groundbreaking discovery, and the toll's role in immunity was so influential that Jules Hoffmann and the other researcher Bruce Butler, I'm not sure about the pronunciation again, so pardon me for that. So they shared a Nobel Prize in 2011 in physiology or medicine, and for the other researcher, he discovered the TLR4 in mammals. So in Drosophila, the stroll pathway is usually paralleled by the IMD pathway, which primarily targets the gram-negative bacteria. Okay, so this is how the PRRs, the idea of PRRs, it was established back in the date in 1990s, that the PRR, which are also referred as pattern recognition receptors. So what are PRR? PRRs are the pattern recognition receptor, which recognize the patterns, the molecular patterns, and they get activated and initiate the immune responses. So the PRR, they are part of the innate immunity, and once they are activated, they are going to initiate the immune responses, the adaptive immune responses. Okay, so innate immunity, so in conclusion, what the Janeway emphasized, that innate immunity, unlike adaptive immunity, it is non-specific, means it cannot recognize the specific antigen, rather it recognizes a broad spectrum of molecules. Okay, and this broad specificity, it is due to the recognition of conserved molecular patterns such as the lipopolysaccharides, such as the peptidoglycans or the beta glucans on the surface of the fungus. Okay, so those conserved molecular patterns which are shared by the pathogens, they will be recognized by the PRRs, pattern recognition receptors, present in the vertebrates or invertebrates, and that is going to activate the innate immunity. So innate immunity is immediate, unlike the adaptive immunity. So it requires no prior exposure or the clonal expansion for the activation, right? So it is immediate, it is broadly specific, and it is also essential, it is also critical for initiating adaptive responses via antigen presenting cells such as dendritic cells. We have discussed this previously in the lectures about cells of immune system. So Drosophila, which lacks the adaptive arm, in Drosophila, it only has the toll pathway, which exemplifies those characteristics, being non-specific, being broadly specific, being immediate, and clarifying the pathogen, specifically the fungus, specifically the bacteria. So this PRR, they exist the pathogen associated molecular patterns, which are referred as PAMPs. So PAMPs are pathogen associated molecular patterns, such as I discussed lipopolysaccharides, peptidoglycans, as well as double stranded RNA. So Janeway only focused on the recognition of the PAMPs by the PRR. So he argued that PRR must exist to detect PAMPs. And then later it was, it was evidently proved by this paper. It showed a genetic demonstration of the existence of PRRs, right? But this idea of PRR recognizing the PAMPs, this was extended later in the date, later in date after by researchers Polly, and they emphasize that not just, PRR, they are not just sensing the PAMs, but they also senses damage associated molecular patterns. So they they recognize pathogen associated molecular patterns alongside damage associated molecular patterns. And this is a really important discovery, because this danger theory extended this into dams, which are host derived signal, okay, that are going to alert the immune system against the internal damage or stress. So whenever there's defense system, right, okay, have an analogy that cops and whatever defense system a country has, it is not just to, tackle the invasion by the enemies or the invaders, but it is also to tackle the, the some consequences of something when it goes wrong in the system, in the country. So it not just takes care of the rebels and the crimes, so it not just take care of the invaders, but it also take care of the crimes and rebels that are there in their country, in that empire, right? In the same way, the immune system not just takes care of the pathogenic attack, but it also takes care of whatever the things are going wrong inside the host. So let's say there's necrosis due to wound, there's tissue damage. So all those tissue damage and necrosis, they are going to, induce the host derived signals, and those host derived signals will alert the immune system against the internal damage or stress. So let's say when there is oxidative stress, or there's tissue damage, so the internal immune system is going to get activated, that now there's something wrong inside the host and we need to tackle those situations. Okay, so DAMs are also recognized by PRR. So this, give a further idea about how it is important to how the PRR are really important, and they are a part of innate immunity, which are going to also initiate the adaptive immunity. So the innate immunity and adaptive immunity, they go hand in hand. So they help each other in the activation. So innate immunity is really important. So this conclusion, Janeway's vision, was realized due to the toll pathway discovery. Okay. So with this, I conclude the lecture. In next lecture, we are going to discuss about the specific types of those PRRs, the toll-like receptor TLRs, the nod-like receptor NLRs, Rigi-like receptor RLRs, ALRs, the CLRs. Okay, so we will be discussing about those different types of PRRs in detail, where they are situated, what's the location, and what are the, the receptors, what are the molecular patterns they are going to recognize. We will discuss about that in detail. And that's it for today. In next class, I'll see you. Till then, bye bye.